Dear students, welcome to this program. Today we are discussing an important topic entitled Genetic Variations. Before discussing in detail our topic, let us first know its objectives. After listening to this lecture, you will be able to describe genetic variation, give important sources of genetic variation, recall the types of genetic variation. Let us begin with Introduction to Genetic Variations. Genetic variation refers to morphological, physiological, cytological, and behavioristic differences between members of the same species or those of different species. They are found in all the characters and in every conceivable direction. Therefore, no two individuals are similar. Sexually reproducing organisms have abundant genetic variations. The variations become fewer if there is intensive inbreeding. Now let us talk about the sources of genetic variations. For a given population, there are 4 sources of variation: mutation, sexual reproduction, genetic drift, and gene flow. Mutations are changes to an organism's DNA and are an important driver of diversity in populations. Species evolve because of the accumulation of mutations that occur over time. The appearance of new mutations is the most common way to introduce novel genotypic and phenotypic variants. Some mutations are unfavorable or harmful and are quickly eliminated from the population by natural selection. Others are beneficial and will spread through the population. Mutation is the raw material for evolution. Had there been no mutation, all the genes would have been present in only one form, and there would have been no evolution and no adaptation to the environment. Mutations are of 2 types: gene mutations and chromosomal mutations. Gene mutations. The most common type of mutational change is the single nucleotide substitution, which changes only a single base pair. Such mutations are also called as point mutations. Single nucleotide substitutions can be divided into 2 types: transition and transversion. Transition is the substitution of one pyrimidine by the other or of one purine by the other. This replaces a GC pair with an AT pair or vice versa. The less common class is the transversion, in which a purine is replaced by a pyrimidine or vice versa, so that an AT pair becomes a TA or CG pair. Sickle cell anemia is an excellent example of a disease caused by a mutation in a gene. The GAG in the 6th codon of the non-template strand of the normal gene changes to GTG. This in turn results in insertion of a non-polar valine residue in the 6th amino acid position of sickle cell alpha globin, where a charged glutamate ought to go. This change in hemoglobin structure can have a dramatic effect on the shape of red blood cells, converting them from disc-shaped cells to sickle-shaped cells, which tend to clog small blood vessels, causing pain and life-threatening crisis. Other common forms of gene mutations include deletions, insertions, and duplications of one or more bases. If the number of bases deleted is not a multiple of 3, then the insertion mutation is also considered a frameshift mutation. Frameshift mutations change all of the codons following the location of the DNA change. Deletions. It involves deletion of one or more bases. A polymorphic deletion variant DF548, which is responsible for cystic fibrosis, is found in the cystic fibrosis transmembrane regulator gene. This variant arose by deletion of 3 base pair at codon 548 in a functionally important domain of the protein, resulting in the loss of the amino acid phenylalanine at this site. Insertions. Just as the name implies, an insertion occurs when nitrogen bases are accidentally added in the middle of the sequence. This throws off the reading frame of the DNA and changes all codons that come after the insertion and therefore completely altering the protein. Huntington's disease is the example of insertion mutation, wherein trinucleotide repeats are inserted into the DNA sequence leading to this disease. Chromosomal mutations. Chromosomal mutations are changes in the number of chromosomes or the structure of a chromosome. Second source of genetic variation is sexual reproduction. The behavior of chromosomes during meiosis and fertilization is responsible for most of the variation that arises in each generation during sexual reproduction. Three mechanisms that contribute to genetic variation are independent assortment, crossing over, and random fertilization. Independent assortment of chromosomes contributes to genetic variability due to the random orientation of tetrads at the metaphase plate. There is a 50-50 chance that a particular daughter cell of meiosis first will get the maternal chromosome of a homologous pair, and a 50-50 chance that it will receive the paternal chromosome. Therefore, the first meiotic division results in independent assortment of maternal and paternal chromosomes into daughter cells. Crossing over produces recombinant chromosomes, which combine genes inherited from each parent. Crossing over begins very early in prophase first, as homologous chromosomes pair up gene by gene in crossing over. Homologous portions of two non-sister chromatids exchange DNA material. For humans, this occurs 2 to 3 times per chromosome pair. One sister chromatid may undergo different patterns of crossing over than its match. When the chromatids separate as chromosomes, the resulting haploid gametes will be genetically unique and varied. Random fertilization. Random nature of fertilization is another source of genetic variation in offspring. Any sperm can fuse with any egg. A zygote produced by mating of a woman and man has a unique genetic identity. All three mechanisms reshuffle the various genes carried by individual members of a population. Third source of variation is genetic drift. Genetic drift describes the effect of chance on populations in the absence of positive or negative selection pressure. Though random sampling or the survival or reproduction of a random sample of individuals within a population, allele frequencies within a population may change. Rather than a male gorilla producing more offspring because he is stronger, he may be the only male available when a female is ready to mate. His genes are passed on to future generations because of chance, not because he was the biggest or the strongest. Small populations are more susceptible to the forces of genetic drift. Large populations, on the other hand, are buffered against the effects of chance. If one individual of a population of 10 individuals happens to die at a young age before leaving any offspring to the next generation, all of its genes will be suddenly lost. In a population of 100, that individual represents only 1% of the overall gene pool. Therefore, genetic drift has much less impact on large populations' genetic structure. Bottleneck effect. Genetic drift can also be magnified by natural events, such as a natural disaster that kills a large portion of the population at random. The bottleneck effect occurs when only a few individuals survive and reduces variation in the gene pool of a population. The genetic structure of the survivors becomes the genetic structure of the entire population, which may be very different from the pre-disaster population. The founder effect. Another scenario in which populations might experience a strong influence of genetic drift is if some portion of the population leaves to start a new population in a new location, or if a population gets divided by a physical barrier of some kind. For example, the Afrikaner population of Dutch settlers in South Africa is descended mainly from a few colonists. Today, the Afrikaner population has an unusually high frequency of the gene that causes Huntington's disease because those original Dutch colonists just happened to carry that gene with unusually high frequency. This effect is easy to recognize in genetic diseases, but of course, the frequencies of all sorts of genes are affected by founder events. Now, the last source of genetic variation is gene flow. An important evolutionary force is gene flow, the flow of alleles in and out of a population due to the migration of individuals or gametes. While some populations are fairly stable, others experience more movement and fluctuation. Many plants, for example, send their pollen by wind, insects or birds to pollinate other populations of the same species, some distance away. Even a population that may initially appear to be stable, such as a pride of lions, can receive new genetic variation as developing males leave their mothers to form new prides with genetically unrelated females. This variable flow of individuals in and out of the group not only changes the gene structure of the population, but can also introduce new genetic variation to populations in different geological locations and habitats. Maintained gene flow between two populations can also lead to a combination of the two gene pools, reducing the genetic variation between the two groups. Gene flow strongly acts against speciation by recombining the gene pools of the groups and thus repairing the developing differences in genetic variation that would have led to full speciation and creation of daughter species. For example, if two populations of a deer species are separated by a mountain range, on rare occasions, a few deer from the western population travel through a narrow pass and become members of the eastern population, thereby changing some of the allele frequencies in the population and promoting gene flow. Finally, let us talk about the types of variation. Because a great deal of genetics concerns the analysis of variants, it is important to understand the types of variations found in populations. A useful classification is into continuous variations and discontinuous variations. Continuous variations. Continuous variations are also called fluctuating variations because they fluctuate on either side of a mean or average for the species. They show differences from the average which are connected with it through small intermediate forms. If plotted as a graph, the mean or normal characteristic will be found to be possessed by maximum number of individuals. The number of individuals will decrease with the increase in degree of fluctuation. The graph will appear to be bell-shaped. Continuous variation is the combined effect of many genes known as polygenic inheritance and is often significantly affected by environmental influences. For example, your height will depend on what genes you inherit and the amount of food you get during your growing periods. You may inherit the genes to make the pigment melanin in your skin, but you will need to expose your skin to sunlight to acquire a tan. In continuous variation, there is no one-to-one correspondence of genotype and phenotype. For this reason, there is little known about the types of genes underlying continuous variation. Discontinuous variations. They are also called saltations or mutations. In discontinuous variation, a character is found in a population in two or more distinct separate forms called phenotypes, without any intermediate stage. Human blood group is an example of discontinuous variation. In the ABO blood group system, only 4 blood groups are possible: A, B, AB, or O. There are no values in between, so this is discontinuous variation. Such alternative phenotypes are often found to be encoded by the alleles of one gene. Similarly, you are either male or female. There are no intermediates. Discontinuous variations are entirely genetically controlled. They cannot be altered by external conditions. Genetic defects such as color blindness, albinism, achondroplastic dwarfism, sickle cell anemia are all genetically controlled and expressed in a discontinuous way. Continuous variation is encountered more commonly than discontinuous variation in everyday life. One area of genetics in which continuous variation is important is in plant and animal breeding. Many of the characters that are under selection in breeding programs, such as seed weight, milk production, arise from many gene differences interacting with environmental variation, and the phenotypes show continuous variation in populations. With this, we conclude our today's lecture. Hope you have enjoyed and understood it very well. Thank you.